Hello students, so this is the continuation of the chapter Cell, the Unit of Life. We have already discussed about the prokaryotic cell. Then what do we saw? We saw that about eukaryotic cell. In the eukaryotic cell we have covered cell wall, right? Then we covered what? Cell membrane. Then we covered endoplasmic reticulum, Golgi complex, isn't it? Thereafter what did we do? We learnt about lysosome. Then we learnt about vacuoles. Okay, so most of these things are already covered in the last class. So moving on with the next cell organelle what we are going to discuss is a very important cell organelle, mitochondria. Okay, so what is mitochondria? So when you see what is mitochondria is a cell organelles which is present in where? In the eukaryotic cell. And what is the function of this mitochondria? You have learned already, it is helping in respiration. They are called as the powerhouse of the cells, because in the mitochondria only the aerobic respiration is taking place and they are releasing huge amount of energy, that energy which is needed for carrying out all our activities. Okay, so that's why mitochondria is called as what? The powerhouse of the cell. Energy is generated where? In the mitochondria. So that is the role of what? This mitochondria. Now when you are looking into the structure of the mitochondria, how the structure look like? So if you are just seeing, they are having two membranes. One is the outer continuous membrane, okay, and thereafter what you can find is that the inner membrane. The inner membrane, when you see, they are not continuous. They fold inwards, go out again, they fold inwards, go out, they fold inwards, like this you can find the constriction is there, inward constrictions are there. Okay, so these are the two membranes which are present in the mitochondria. This is intermembrane space. Okay, so this is space we call it as for the intermembrane space. Okay, thereafter you can find another one space is also present, the cavity present over here. This cavity we call them as, we are calling them as what? We are calling them as so we are calling them as what? The matrix. What do we call them? We call them as the matrix. So the cavity which is present, we call it as what? We call them as the matrix. Now when you are seeing this matrix, in the matrix you can find much amount of fluid is present, and this inner membrane is folding inwards, isn't it? So this inward folding part, we call it as what? We call them as cristae. What do we call them? Cristae. So moving on to the structure, you are having an outer membrane, you are having an inner membrane. Intermembrane space is present in between, which is fluid filled. Again there is a cavity inside which is fluid filled. Then you can find the inner membrane is having infoldings. That infoldings we call it as what? We call them as cristae. And in this inner membrane you can find huge amount of enzymes are located. Okay, some particles are located which are helping in the process of what? They are helping in the process of respiration, carrying out what? Respiration. Okay, and one more thing what has to be noted is that this mitochondria is containing ribosomes. Along with that they also have genetic material, DNA. What is present in them? The DNA. So what is the characteristic feature of mitochondria then? If a DNA is present, what they can do is that mitochondria can undergo division by itself. The cell need not divide to increase the number of mitochondria, because they are having what? The DNA. That's why they are self-replicating. Self-replicating means what? Whenever they are feeling that the cell needs, my body needs more amount of energy, at that time you can find this mitochondria will divide. The DNA will form its duplicate copy. The DNA will divide first, and if from one mitochondria, how many mitochondria will be formed? 2. From this 2 mitochondria, how many will be formed? 4. Like this, the process of division we call it as fission. What is that process called? Fission. With the help of this fission you can find the number of mitochondria increases. Okay, so whenever there is need for much amount of energy production, mitochondria will self-replicate or self-duplicate itself, okay, within the cell. That's why they are called as self-replicating bodies, and this characteristics you can also find in the chloroplast. Chloroplast, you know, plastid, one of the part is what? The chloroplast. What is the function of chloroplast? The chloroplast contain chlorophyll and they help in what? Photosynthesis. Now here when you are seeing, the chloroplast is also having what? This DNA, and they are also what? Self-replicating, means they can divide to increase in number in the plant cell. So from 1 chloroplast 2, from 2 to 4, by fission they can also divide. Okay, so name the two self-replicating or self-duplicating body or cell organelle. If it is asked, what are the two? One is mitochondria and another one is chloroplast. Hope so it's clear. This is regarding a general discussion about what this mitochondria. Now we will learn in detail. The number of mitochondria per cell is variable. I told you they can divide and they can increase, they can means number may be less. They are sausage shaped, means what, they are somewhat cylindrical in shape, isn't it? So when you are seeing this structure of this mitochondria, they are somewhat like a cylinder, right? Yes, so that is next point. Thereafter, what is the diameter? When you see, it's 0.2 to 1 micrometer, and the length, if you are seeing, so that is diameter and another one is the length. So when you are seeing the length is about how much? It is 1 to 4.1 micrometer. Okay, and it is double membrane. I told you they are made up of two membranes, right? Outer and the inner membrane, and dividing its lumen distinctly into two aqueous compartments. There is two fluid filled structure. One is the outer compartment and the inner compartment. The inner compartment we call it as what? The matrix. So when we were drawing, we have already drawn, there are two compartments, two fluid filled compartments. One is the outer compartment as well as another one is the inner compartment. So which one is the outer compartment? This is the outer compartment. Which is the inner compartment? This is the inner compartment. Okay, the inner membrane folds into infoldings called as what? Cristae. That infolding, what do we call? We call them as cristae. So this is the infolding which we call it as the cristae, and they are increasing the surface area, so much amount of enzymes, so aerobic respiration is carried out mainly there. They produce cellular energy, and the energy is formed in the form of what? ATP, stored in the form of a chemical energy called as what? ATP. Okay, so the heat energy which is generated during the process is converted into a chemical energy called as ATP, adenosine triphosphate. Okay, what is the full form of ATP? Adenosine triphosphate, and that heat energy which has been released is converted into ATP during respiration, and it is stored in the cell. Okay, that's why we call them as what? Powerhouse of the cell. So whenever energy is needed, this ATP is broken down. When the ATP is broken down, the captured energy is released, and that energy is used for carrying out our work. Now when you are moving on to the rest characteristics, matrix, there is a DNA. I told you they have what? A single circular DNA. That's why they are self-replicating. They can divide. Okay, they have RNA molecules, they have ribosomes and 70S. I told you, like prokaryotes, here mitochondria is having what? 70S. But when you are seeing the cytoplasm, it is what? 80S. Eukaryotic cell, they are having 80S. Okay, and the components required for the synthesis of proteins are all present where? In the mitochondria. And this mitochondria, they divide by a simple division called as what? Fission. What is the process? Fission. So this is the structure. You can find in a intermembrane space. Yes, thereafter you can find the green part is the matrix, cristae is the convulsion, inner membrane and outer membrane. Fine. Now this is again cristae, mitochondrial DNA. So this is the circular DNA which is present. This is matrix, intermembrane space is present, outer membrane, inner membrane, intermembrane space, ribosome is present, cristae is present. Okay, so this is regarding walled. These are stacked particles which are playing a major role in what? Aerobic respiration. With this, we are moving on to the next topic. So hope so mitochondria is clear, the structure as well as the function, and why it is called as powerhouse of the cell. Now moving on to plastids. So plastids, there are two. One is plasmid, extra chromosomal DNA present in prokaryotic cell. We call it as plasmid, MID, isn't it? This is plastid, present in eukaryotes. What is plastid? Plastids are found only in plant cells, where you can find plant cells. Okay, and when you are seeing this, plastids are the structures or the parts or the cell organelles which is imparting color to various parts of the plants. Okay, now this colors, when you are seeing based on them, the plastids can be classified. Okay, plastids can be classified into how many types? It can be classified into 3 types. Okay, based on the type of pigment, whether it is present or whether it is not present. If present, what is the type of pigment present? Based on that, it can be classified into 3 major categories. So what are the 3 major categories? One is the chloroplast, second one is chromoplast, and another one is what? Leucoplast. Chloroplast, as the name suggests, they contain chlorophyll. Green in color, they are helping in what? Photosynthesis. Chromoplast, when you are seeing chromo, colored, okay. Colored plasmids, other colors of the plants, like you have orange, yellow, violet, isn't it, red, all these colors are also present in the plant, flowers everywhere, or different parts you can find, fruits and all, when you are finding they are having different colors. Why do they have? Because of the presence of the chromoplast. So other than green, the other colors are represented by whom? This chromoplast, like xanthophyll, they have different pigments like xanthophyll, carotenoid, isn't it, fucosanthin, like that you have learned a lot, right? So that is your chromoplast. Next, moving on to the next is your leucoplast. What is this leucoplast? Colorless plastids, yeah. They do not have any color. They do not impart any color. So what is the function of them? They are acting as storage tissues. What they are acting as? Storage, okay. So three types of plastids, what are they? One is chloroplast, second one is chromoplast, and third one is leucoplast. They are colorless, they are colored, this is green colored, okay. Now, chloroplast contain chlorophyll, okay, for photosynthesis. Chromoplast contain fat soluble like carotenoids, xanthophyll, keratin, all these things. They are imparting different colors. Leucoplast are what? Colorless plastids. They are of various shapes and size, and they are helping in storage. Now what they are storing? Based on what they store, they are of three types, okay. So what are they? Amyloplast, if the storage is starch, carbohydrate, then it is amyloplast, okay. If they are storing something like fats, okay, fats, lipids or oils they are storing, in that case it is elaioplast, okay. And if they are storing proteins, then we call it as aleuroplast, okay. So these are the three different types of what? Leucoplast, based on their storage. Amyloplast is storing what? Starch. What is elaioplast is storing? They are storing fat and oil. What is aleuroplast is storing? They are helping in the storage of protein. So these are the three different types of what? This leucoplast, right. Now when you are moving into the structure of this chloroplast, how the structure of the chloroplast is, listen carefully. So when I am drawing a chloroplast, again they are also having what? They are also having two membranous covering, okay, outer membrane as well as the inner membrane. So they are having how many membranes? Two membranes, outer membrane as well as one inner membrane, okay. And when you are seeing, both are continuous only. Inside that fluid is present. This fluid, what is present inside, we call it as stroma. What do we call them? Stroma, okay. There are disc-like structure present, and they are arranged one after the other like this, okay. So this disc-like structure, one single disc, we call it as thylakoid. What do we call them? We call them as thylakoid, okay. And when all these thylakoids, when you are seeing, they are joined together, arranged and stacked all together, we call it as grana. What do we call them? We call them as grana, okay. Now this one grana is connected with the another grana through bridges, okay. So this is one grana, another grana, if you think, so they are connected by bridges, okay. So that bridges, we call it as stroma lamella. What do we call them? We call it as stroma lamella, okay. So this is regarding the structure. Now what is the function of this chloroplast? When you are seeing chloroplast, you know they are helping in what? Photosynthesis. Photosynthesis, a part of photosynthesis takes place only in the presence of light. We call it as light reaction, okay. And another one, it takes place most of the time, okay. Even in dark also it takes place. That's why we give the name as what? Dark reaction. So stroma, when you are seeing, there mostly dark reaction is taking place. And for the light reaction to take place, what is needed? Chlorophyll, okay. Chlorophyll is needed, and light is also very important. That's why the name is what? Light reaction. The light reaction is taking place in the grana, because in the grana membrane only what you can find? You can find the chlorophyll, and the light reaction is taking place in the grana, dark reaction is taking place in the stroma. So photosynthesis consists of two reactions, one is the dark and another one is light. Light takes place only in the presence of light, dark can take place at both the sides. If it is light is present, no problem. If it is dark also, it's not a problem, they will take place, okay. So that is regarding your structure of what? The chloroplast, right. So chloroplast, double membrane, Space limited by inner membrane of the chloroplast, we call it as what? Stroma. Done. Flattened membrane sacs are present. So, these flattened membranous sacs which is present is called as what? Thylakoid, right. So, this fluid filled space we call it as what? The stroma. Flattened membranous sacs we call it as thylakoid. Thylakoid are arranged in stacks like piles of a coin, isn't it? So, they are arranged like piles of a coin. Coins are arranged one after the other, just like that. That one we call it as what? Grana. What do we call them? We get called grana. And you can find flat membranous tubules which are connecting the thylakoids, isn't it? Of different grana, they are connecting. So, this is connecting, this is connecting, this is connecting, right. So, this connecting parts we call it as what? Stroma lamella. What do we call them? Stroma lamella, okay. The membrane of the thylakoid encloses a space called as lumen. So, this membrane, when you are saying thylakoid, one disc if I am taking, they are having what? A space in between them, fluid filled space. That cavity we call it as the lumen, okay. The stroma of the chloroplast contains enzyme required for the synthesis of carbohydrate, protein, everything is being synthesized. As well as I told you, they are having what? DNA. Like mitochondria, they have DNA and they are helping in what? Division. So, chloroplast can divide itself within a plant cells, okay. As well as they have ribosomes. Now, chlorophyll pigment, where it is present in the thylakoids. In the grana only you can find what? The chlorophyll pigment. And the ribosomes of the chloroplast are 70 years. I told you it is 70 years in prokaryotes as well as in the eukaryotic cell. Inside the mitochondria and inside the chloroplast, it is 70 years. But when you are seeing a eukaryotic cell in the cytoplasm, what it is? The ribosome which is present, that is 80 years, okay. These things we have already discussed, right. Now, we are moving on to the next organelle called as what? Ribosomes. Hope chloroplast is clear. Chloroplast and mitochondria are very important. You have to learn the structure of mitochondria, structure of chloroplast, function of mitochondria and function of chloroplast in detail. It's a 3 mark or a 5 mark question, okay. If it goes with the diagram, it's a 5 mark question. So, learn properly. Now, moving on to the next part, it is ribosomes, okay. Ribosomes are the glandular structure. So, ribosomes, when you are seeing just like this round bodies which are present inside the nucleus, as well as you can find it in the endoplasmic reticulum, you can find it in the cytoplasm, okay. It was observed under electron microscope as dense particles. So, these dense particles was observed by whom? George Palade was the one who observed it in 1953. They are composed. When you are saying the composition, what it is made up of? It is made up of RNA. What it is made up of? It is made up of RNA, ribonucleic acid, and they have protein. They are also having what? Protein. So, what is the composition of ribosomes? They have RNA plus protein. But when you are seeing this ribosomes, they are not having a membrane. What is not present? A membrane is not present. So, the RNA and protein are present just like that, somewhat spherical in nature, but there is no membranous covering. Eukaryotic ribosomes are 80 years, while prokaryotic ribosomes are 70 years. I told you 80 years, when you 80 years, how it is coming? It is like you have 60 plus 40, isn't it? And when it is coming to your 70 years, how you are getting? You are getting it as 50 plus 30, isn't it? So, 70 years is 50 plus 30. I told you a part of it is what? Immersed. So, this becomes common for both, because of which you can find 50 plus 30 instead of 80, it becomes what? It becomes 70, right. So, the S is standing for sedimentation quotient. I told you coefficient when you are seeing centrifugation in that process, Sedgwick's unit, okay. So, sedimentation coefficient, it indirectly is a measure of what? Density and size, how much big, how much dense it is. Both 70 years and 80 years are composed of 2 subunit, isn't it? One light unit, light chain, and another one is the heavy subunit. So, both, whether it is 80 years or 70 years ribosomes, they are having 2 units, one light unit and another one is heavy unit. The next thing what we are going to discuss is cytoskeleton, which are tubule like structures, okay, tube like structures which are present in the eukaryotic cell. We call it as what? The cytoskeleton can be present in prokaryotes as well. This cytoskeleton is an elaborate network of filamentous proteinaceous structures present in what? The cytoplasm. So it's a filamentous structure which is made up of mainly protein in the cytoplasm, highly distributed. We call them as what? Cytoskeleton. Now when you are seeing the cytoskeleton, if you are looking at under microscope with radiations, light radiations, you can find these as like you can find the fibers, right? The what is that? The tubes running out. So it's a filament like structures, isn't it? So this one we call it as what? We call them as the cytoskeleton. Here we are going to discuss about the filaments, two filaments. One is the cilia and the flagella, and another one is the centrosome. Okay. So two organelles we are two parts we are going to discuss. First part we are going to discuss about the structure of your cilia and flagella, followed by we will learn the structure of the centrosome. Okay. Now you can find this is a section of cilia and flagella. Cilia and flagella, you know how it will be. Like for example, bacteria or any filament, if you are taking, cilia and flagella are filamentous, isn't it? So if it is small, it is cilia, and if it is much bigger, then it is flagella, isn't it? And what is the cilia flagella doing? Cilia, when you are seeing, they are helping in movement, isn't it? Maybe they are helping in moving the fluid, or lashing and moving the substances from one place to another, or the cilia itself is helping in the moving of an organism. If it is like prokaryotes and all, when you are seeing, they are helping in the movement of the organism. But when you are seeing this flagella, what they plainly play, they play a role in what? Locomotion, moving from one place to another. Okay. Now if you are taking a cross section of this flagella or cilia, how it will be looking like? That is the discussion which is discussed in your text. Not that important anyhow, we will be discussing what how does it look like. Okay. Now moving on to the structure. See, this is one cilia, right? So this is one flagella. Imagine this is one flagella, right? Now what do you mean by cross section? Cross section means with the help of a blade I am cutting. Yes. Then I am taking a thin section. This section I have taken. The small section, what I am taking it out, this one we call it as what? The cross section, right? This one I am keeping it under the microscope. So first I have cut the tip, thereafter with the blade I have made a small cut, and I am taking a section of the same. That one we call it as what? CS, cross section. Okay, CS has been taken. Now this cross section, if you are viewing in detail what and all it is made up of, that's what we are going to see. Okay. So cross section, when you are seeing of this flagella, they are made up of many tubules, microtubules, proteinaceous filamentous structures called as what? Tubules. Okay. So how many tubules they are made up of when you are seeing? So this is one flagella. Imagine this is one flagella. Okay. So in the cross section, when you are seeing how they are made up of, they are made up of two central tubules. How many tubules are there? Two central tubules. Thereafter you can find how many tubules are on the periphery? 9. How many? 9. So 1, 2, 9, that to doublet. 3, 4, 5, 6, 7, 8, 9. So like this, how many tubules are present? 9 tubules are present. Okay. So how we are going to see this? So one, if I am taking a cross section, the tubules, how it is represented, that's what we are going to see. Okay. So you see, so I have kept the cross section over here, right? So the center you can find two tubules present. So this is one tubule and this one is another tubule, right? So how many tubules are present? Two tubules. One is this one and another one is this one, right? They are connected by what? A bridge. What they are connected by? A bridge, right? The central part, we call it as the central sheath. What do we call them? Central sheath. So I have taken a cross section and I am seeing it under the microscope. It will be looking like this. Thereafter, so central two microtubules are present connected by a bridge, and this portion we call it as the central sheath. Now moving on to the next part, what we are going to discuss is that the periphery, or towards the periphery. Okay, so which one is the periphery? This is the periphery, isn't it? So on the periphery, when you are seeing, there are 9 doublets, 9 peripheral tubules. Okay, so microtubules 9 are there, each are made up of 2 fibers. So 1, 2, 3, 4, 5, 6, 7, 8, 9. That's why this sort of arrangement we call it as 9 plus 2 array. What do we then? we call them as 9+2 array. This peripheral microtubules are doublets. Doublets means what? They are made up of two sets of proteins in structures. Okay. So each are doublets, isn't it? They remain in pair. So that's why it is called as what? The doublets, right? Now this peripheral tubules, when you are seeing, they are connected to the central sheath. So this is the central sheath by a spoke, like a cycle you can find in the center, isn't it? So when you are seeing the cycle tire, in that case you would have seen that the cycle tire, how it will be, you will be having the cycle like this, thereafter the center will be here, you have spokes like this, isn't it? The wheel when you are seeing, they have spokes like this, isn't it? So like that, the peripheral tubules, when you are seeing, they are connected to the center by this connecting structures. We call it as what? The radial spoke. What do we call them? We call them as a radial spoke. Okay. And each this microtubules, peripheral microtubules, when you are saying, they are also connected. Yes or no? Yes, they are connected. Okay. How they are connected? They are connected by interdoublet bridge. What they are connected with? They are connected with interdoublet bridge. Okay. So you can find this is the interdoublet, yellow color round protein structures you can find them, isn't it? So that one, this is interdoublet bridge, this one, okay. So this is interdoublet bridge. So what is the function of this? They are connecting this peripheral tubule with this peripheral tubule, okay. So this is the structure of what? A cilia. What is this? A cilia of flagella structure it is, okay. Now we will discuss in detail. Moving on to the cilia. Cilia and flagella are hair-like outgrowths. Cilia are smaller structure, flagella are comparatively longer. They are helping in the movement of either the cell, they are helping in moving, or the part of the fluid they are helping in moving, okay. And flagella, I told you, for cell movement. They are so what study we did is electron microscopic study, means under the electron microscope very in detail we are learning the structure. When you are seeing, they are covered with the plasma membrane. Yes, the outer membrane is what was? It was the plasma membrane, okay. Plasma membrane. This core is called axoneme. The central sheath, I told you, na, that one we call it as axoneme. Possess a number of microtubules, not the central sheath, the whole part, okay. So this part, the whole part, we call it as what? The axoneme. It contains a number of microtubules running parallel to one another. Each and every one is running parallel to one another. So 2 central, the rest 9 peripheral, all are running what? Parallel to one another over the long axis, right. Now axoneme has 9 pairs of doublets. I told you 9 pairs of what are there? Doublets are there, isn't it? Like this 9 pairs are there, and a pair of what? Centrally located microtubule, 1 pair center, right. Such an arrangement we call it as 9+2 array. The central tubules are connected by bridges and they are enclosed in a sheath called as central sheath, which is connected to one of the tubules of each of the peripheral doublet. So by the radial spoke, these doublets are being connected, isn't it? And how many radial spokes will be there? Since 9 doublets, so 9 radial spokes will be there. And the peripheral doublets are also interconnected by the linkers. So you can find there are intermediate linkers which are connecting the two. So this is one doublet and this one is another doublet, means you can find the peripheral linkers are connecting what? This one, right. So that is another one characteristic of what? This structure which are present in the flagella, right. So this both the cilia as well as the flagella, when you are seeing, they are emerging from a centriole like structure, centrosome like structure called as what? Basal bodies. So from where they are coming, these tubules are originating, there are special protein structure present, we call it as what? The basal bodies. Now moving on to the structure of centrosome and centrioles. See, centrosome or centrioles you have learnt in your animal cell, right. So this one is the centrosome or the centrioles which you have learnt where? In the animal. So you can find this is the centriole, right, in, yeah. So this is what? This one we call it as the centriole. So how the centriole looks like, okay, that's what we are going to discuss now. The same like cilia, flagella only, they are also having somewhat similar structure, but a bit differences are there. So that one we will see, okay. So centrosome or centrioles, when you are seeing, they are perpendicular to each other, okay. So one centrosome is made up of 2 centrioles. How many centrioles? 2 centrioles. And these centrioles, when you are seeing, they lie perpendicular to one another. So when you are looking into one of the centrioles, okay, so what is the structure of this centrioles when you are seeing? So the centrioles, when you are seeing, they are surrounded by amorphous centriolar, what is that, material with some sheath like means somewhat sticky substances will be present. With that only they are surrounded. And when you are seeing the centrioles of the centrosomes are lining what, perpendicular to one another. So what is centrosome? Centrosome is a structure which is present only in animals. Now the centrosome, when you are seeing, it is made up of 2 centrioles. How many centrioles? 2 centrioles. And these 2 centrioles, they lie what, perpendicular to one another, and they are being covered by what, an amorphous pericentriolar material. Now the centrosome centrioles, when you are seeing, what is this structure of the centriole? When you are seeing, it's somewhat similar to that of the flagella. They also have 9 peripheral tubules, isn't it? But it is not doublet. It is how many? 3 are there, isn't it? So they are triplets. What they are? Triplets in nature. Okay. So they are having 9 evenly spaced peripheral fibrils of tubulin. Okay. And each of the peripheral fibril is a triplet. I told you, not like flagella. Here it is triplet. 3 are there. The adjacent triplets are also linked. Here also you can find the triplets are all linked with one another. Yes or no? Yes, they are linked with one another, right? So I will just change the color so that it becomes easier. Fine. So they are all linked with one another. You can find one triplet is linked with that of the other triplet. Like there also linkers were present in flagella. Here also what is present? The linkers are present. Okay. And the central part of the centriole is proteinaceous. We call it as hub. The central part we call it as what? We call them as the hub, 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 which is connected with the tubules. So they are all connected with the tubules, isn't it? So they are all connected with the tubules with the help of radial spoke. So this one we call it as what? Radial spoke. How many are there? 9 are there. So automatically 9 peripheral fibrils are present, means here also how many will be present? The radial spokes will also be 9 in nature. Okay. All are made up of what? All these substances are made up of different types of proteins. So centrosome and centriole is a structure which is present only in animal cell, and they are having structures similar to that of the flagella, as they are tubule like structures, okay, fiber like structure. That's why they are like that, right? So this is regarding your centrosome and centriole. Hope it's clear. Now we are moving on to the last part of the chapter, and that is nucleus. What we are going to learn? Nucleus. With this we will be winding up the chapter. Now nucleus, when you are seeing, I will discuss about the nucleus first, then we will see line to line what and all has been discussed. Okay. So nucleus. So nucleus, if you are seeing this nucleus, what it is made up of? The nucleus is present mostly in the center, isn't it, of an animal cell, and if it is a plant cell, it may move towards one of the side, it may be moving because of the presence of a large vacuole. The nucleus is made up of 2 coverings. How many coverings are there? 2 coverings. Okay. So then blue, I think black will be better. Let me change it to black. Yes, fine. Now if you are seeing, 2 membranes are present. Okay. Outer membrane and inner membrane, right? This outer membrane and inner membrane is not continuous. What is present in between? You can find spaces are present. Okay. So the spaces, we call it as nuclear pore. What do we call? We call this one as nuclear PORE, pore. Okay. Inside the nucleus. So when I am drawing, how I have to draw? I have to draw like this. This would be better, isn't it? Spaces are present. So between the nucleus, you can find spaces are present, right? So this space is bigger, but usually it is very small. Okay. So this is regarding your nuclear pore is present over here. Okay. And so the nuclear membrane is not continuous. In between it is disrupted by holes, nuclear pore. Inside the nucleus you can find something is filled. What is filled? It is filled with the fluid. Name of the fluid is nucleoplasm. What do we call that? Nucleoplasm. Like the cell is filled with what? Cytoplasm. Here nucleus is filled with nucleoplasm. In the nucleus you can find a round, somewhat darker spherical structure. We call them as nucleolus. What do we call them? Nucleolus. Okay. So, nucleoplasm, fluid. In the fluid only all the activities of the nucleus, what has been controlled, is been controlled by whom? This nucleoplasm. And what is this nucleolus doing? Nucleolus is a site where you can find RNA synthesis as well as ribosome synthesis is taking place. They are playing a major role in the synthesis of RNA and ribosome. Thereafter, in the nucleus, you can find a main structure present. What is the structure? The DNA, genetic material, chromatin. What do we call them? Chromatin. So, this is the nucleus. Inside that you can find what? DNA. So, this is DNA. DNA, we call it as, they appear as thread-like structures. Chromatin, we call that chromatin fibers, chromatin threads. Chromatin threads, when they are dividing, they condense to form chromosome. And this chromosome, inside that only you can find the genetic material, what is present? The DNA is present. In detail about the DNA as well as the structure of the nucleus, you will be learning in your class 12. Okay. So, this is regarding what? Your nucleus. Hope it's clear. Now, the points missed out, we will be learning one by one. Okay. Fine. Nucleus. Nucleus is a cell organelle and it is been discovered by whom? Robert Brown. The material of the nucleus stained by the basic dyes was given by the name chromatin. When you are staining it, the things are getting stained. Okay, thread-like structures are getting stained, and the name was given for the stained material present in the nucleus as chromatin by Fleming. Okay. So, interphase means when this nucleus is not dividing, you can find the nucleus, the DNA, the material will be present in the thread-like structure. Those fiber-like structures we call it as chromatin. Okay. And there is a special structure called as nuclei. I told you nucleolus. Thereafter, nuclear envelope is the covering, consists of two parallel membrane with a space between. I told you in the nucleus how many membranes are there? Outer membrane and inner membrane. In between there is a space. Okay. That space we call it as what? The perinuclear space. The outer membrane usually remains continuous with the endoplasmic reticulum and also bears ribosomes. So, this outer membrane, when you are seeing, they are in continuation with what? The endoplasmic reticulum. You can find endoplasmic reticulum, right? So, will be present. So, when you are looking into the diagram of the plant or the animal cell, in that you would have seen that outer membrane is always in continuous with that of the endoplasmic reticulum. So, this is the nucleus and this one is the endoplasmic reticulum. So, outer membrane is in continuous with what? The endoplasmic reticulum. So, that is one of the characteristic feature of what? This nucleus, right? Now, next is your this nucleus. They also bear what? Ribosomes. I told you they are called as what? Rough endoplasmic reticulum. At a number of places, nuclear envelope is interrupted by minute pores. I told you, what do we call them? Nuclear pores. What do we call them? Nuclear pores. So, messages from the nucleus has to go to the cytoplasm through the endoplasmic reticulum or through any other source. So, through the pores only the messages is moving, especially what is going? RNA, protein molecules which is synthesized, is being distributed from the nucleus to the cytoplasm by this nuclear pore. Okay. Some mature cells, when you are seeing some cells, they do not have nucleus. Very important, one more question. Erythrocytes, RBCs, sieve tubes in case of plants. Sieve tubes, you would have learnt phloem. What is it? Phloem tissue having sieve tubes. That cells, they do not have nucleus. Okay. They do not possess a nucleus. Erythrocytes are not having nucleus. Why? Because if the nucleus is present, the erythrocytes will have what? Somewhat spherical structure, so they cannot easily move. They are biconcave, right? So that they can move easily within the capillaries. If the nucleus is present, they will somewhat become what? Spherical in nature, occupy much space. That is one difficulty. The nuclear matrix, we call it as fluid filled part, nucleoplasm. Here only nucleolus and chromatin are present. Okay. Nucleoli, spherical structure, and they are not membrane bound, they do not have a membrane, and there is ribosomal RNA synthesis is taking place. Larger and more numerous nuclei present in cells actively carrying out what? Number of nuclei when you are seeing are larger, bigger in size, where the protein synthesis is more. Okay, much amount of nuclei will be present. By the nuclei is present where the protein synthesis is more, because for protein synthesis RNA and ribosomes are needed, and this RNA and ribosomes are produced by whom? This nucleoli, isn't it? Yeah. Now chromatin, when you are seeing, one is chromatin, I told you inside the nucleus you are having chromatin threads. This chromatin threads, when you are seeing, when they are dividing, they are condensed into rod like structure called chromosomes. Now this chromosomes and chromatin, when you are seeing, they are made up of what? They are made up of DNA. What they are made up of? DNA, plus they are having some proteins. The proteins can be histone proteins or non-histone proteins. Sometimes some amount of RNA will also be present in the chromatin or the chromosomes. Okay. Each chromosome has a primary constriction. So when you are looking into the structure of the chromosome, okay, so always chromosome appear in pair. One is from that of the maternal side, another one from the paternal side. So this chromosomes, when you are seeing, they are having a constriction in the center. So constriction in the center, you can find they are having a constriction. This constriction, constricted part, we call it as the centromere. What do we call them? Centromere. Just in the centromere you can find a disc like structure present, green color structure you can see. That structure we call it as what? The kinetochore. So this is the structure of what? A chromosome. So what is a chromosome? Chromosome is a structure which is present inside the nucleus. This chromosomes only contains the DNA which is controlling all our characters. Now when you are seeing this chromosome, it is having a constriction in the middle. That constricted part we call it as the centromere, and in the centromere you can find disc like structure present on either sides, that we call it as what? The kinetochore. Okay. So this is regarding a structure based on the position of the centromere. Okay, very important 2 mark. Based on the position of the centromere, how many types of chromosomes are there? There are 4 types. How many types are there? 4 types. One is metacentric, submetacentric, acrocentric and telocentric. Listen very carefully, what is the difference? If the centromere, that is the constriction is present, okay, where the constriction is present? The constriction is present in the center, okay, then we call them as what? Metacentric. If the constriction is present not in the center but to some of the side means what? This is metacentric, you see, equal, this side also, the length is somewhat equal. But here when you are saying it's submetacentric, one is somewhat bigger, the constriction is away from the center, a bit away from the center, then we call it as submetacentric. Okay. So in this, one will be a bit longer than that of the other end, isn't it? The other rod. And when you are seeing acrocentric, here the centromere, when you are seeing, the centromere is present towards one of the end, isn't it? So here how it is present? It is present towards one of the end. So towards this end it is present. And one is much longer and another is very small. Other type of chromosome is here telocentric. The centromere is present at the tip of the chromosome. Okay. So then we call it as what? Telocentric. So based on the position of the centromere, how many types are there? 4 types. If the centromere is present in the middle, metacentric. What do we call them? Metacentric. If centromere is present a little bit away from the middle, we call it as submetacentric. If the centromere is present nearer to that of one of the end, acrocentric. And if the centromere is present towards the end, we call it as what? Telocentric. Yeah. So this is regarding your structure of a nucleus. In this nucleus, you have to learn about the 2 nuclear membrane, you have to learn about nucleoplasm, nucleoli, chromatin, structure of chromosome and the different types of chromosome based on the position of centromere. With this, the chapter is getting over. Hope you would have understood the concepts. So we will meet in the next class soon. Thank you.